Welcome to Learnpedia, the ultimate JEE and NEET prep tool that is currently being used by over 20,000 aspirants. Now let's see if you can answer this important question. If you think you got the answer, then post it in the comment section below. To understand the concept behind this question, watch the video that follows.
Light falls on chloroplast. Chlorophyll molecules get excited by absorbing the light energy and they transfer this energy to the reaction center. When photosystem 2 is excited, the energy goes to P680, the reaction center of photosystem 2. From P680, the emitted electron is received by a non-chlorophyllous pigment called pheophytin. From pheophytin, the electron goes to plastoquinone. That means pheophytin is oxidized and plastoquinone is reduced. This plastoquinone, when it receives the electron, it is reduced to plastosemiquinone. Always the electron is accompanied by a proton. Plastosemiquinone is further reduced by accepting one more electron accompanied by a proton, and it is converted to plastoquinol. Plastoquinol transfers the electron to cytochrome B and get itself converted to plastosemiquinone. In this process, the proton that is accompanying is pumped into the lumen. The plastosemiquinone receives one more electron and gets converted to again plastoquinol. This time the plastoquinol conveys the electron to cytochrome F, not B. From cytochrome F, the electron goes to a copper containing electron acceptor called plastocyanin. The accompanying proton is pumped into the lumen. The cyclic transfer of electron starting from plastoquinone is called quinone cycle. Plastoquinone, by receiving an electron, it is converted to plastosemiquinone. By receiving another electron, it becomes plastoquinol. From plastoquinol, when electron goes out, it again becomes plastosemiquinone. By receiving another electron, again it becomes plastoquinol. During this cycle, for every electron transfer, 2 protons are pumped into the lumen. When the photosystem 2 loses one electron, this reaction center gets now positive charge. In the lumen, water breaks up into hydrogen and half O2. From this hydrogen, the electron goes to P680 and its loss of electron is compensated by the electron emanating from the hydrogen that is derived from water. So, for every one water molecule, 2 electrons are generated, and when these 2 electrons go into the quinone cycle, 4 protons are pumped into the lumen. Already 2 protons are there in the lumen derived from water. All these protons that are pumped into the lumen are available in the stroma. These protons only go into the lumen. So in this way, the last electron by PS2 is compensated from water, and this electron through cytochrome F goes to the plastocyanin. Now the same is repeated with the photosystem 1, where the reaction center is P700. When light falls on the photosystem 1, the electron is shot out. The loss of the electron from P700 is compensated from the plastocyanin. This electron goes to a substance called ferredoxin reducing substance, and this electron is transferred to ferredoxin, and from ferredoxin it goes to NADP, and NADP now gets converted to NADPH2. So by the utilization of one water molecule, one NADPH2 is produced and 6 protons accumulate in the lumen. In the lumen, the proton concentration builds up, resulting a lot of potential difference between the lumen and the stroma. The thylakoid membrane is impermeable to protons, but there are some passages in the membrane, they are called coupling factors, present in non-appressed regions. The coupling factor has 2 parts. They are, one, a hydrophobic membrane bound portion called CF0. This is called stalk. And 2, a part which comes out into the stroma called CF1. This is head. The CF0 stalk acts as a proton channel, whereas the CF1 is the catalytic site. This is the site for the synthesis of ATP. It is generally estimated that when 3 protons moves down to the coupling factor, the energy released is enough to add 1 inorganic phosphate to ADP, leading to the production of ATP. When you look at the photosynthetic equation, that is 6CO2 plus 12H2O in the presence of light and chlorophyll, we get 1 molecule of hexose and 6 molecules of oxygen is liberated and 6 molecules of water are regenerated. So for every glucose molecule or hexose molecule produced, there is utilization of 12 molecules of water. For the utilization of 1 water molecule, there is the production of 1 NADPH2 and 2 ATP molecules. For 12 molecules of water, 12 molecules of NADPH2 and 24 molecules of ATP are produced. This forms the assimilatory power, that is, these products are used in carbon assimilation, that is, dark reaction. Synthesis of ATP utilizing the light energy is called photophosphorylation. Photophosphorylation was first suggested by Arnon in 1953. Photophosphorylation by proton translocation was suggested by Peter Mitchell, and it is called chemiosmotic model. For proposing this model, Mitchell was awarded Nobel Prize in 1978. The electron transport so far we have studied is non-cyclic. That means the electron that is released from photosystem 2 never returns to the same place. So it is called non-cyclic photophosphorylation. In addition to non-cyclic photophosphorylation, there is another pathway called cyclic photophosphorylation, and it involves only photosystem 1. It was first demonstrated by Frankel in isolated chromatophores of a bacterium called Rhodospirillum rubrum. In this process, electrons from P700 go to pyridoxine, and from pyridoxine the electrons go to cytochrome B6, and through plastoquinone they go to cytochrome F, and from cytochrome F they go back to P700 through plastocyanin. As quinone cycle operates between cytochrome B and cytochrome F only, for each pair of electrons only 4 protons are added to the lumen. In this cyclic photophosphorylation, no NADP is produced. Water is not involved. No liberation of oxygen. That means no photolysis.
Hey there, hope you understood the concept. Here's the answer to the question that was asked in the beginning. Found this video useful? Hit the like and share buttons to enjoy more such videos. LearnPedia's JEE and NEET prep tools contain over 4,000 videos and over 20,000 questions. You can access them online through our website or offline through an SD card or a pen drive. To buy now, visit www.learnpedia.in. You can also try a free demo of the product before buying.